Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Amni Husna binti Abdul Aziz, and my title will be Evaluation of Antimicrobial and Hemolytic Activity of Pigment Extracted from the Marine Rhodotorula mucila. Introduction. Carotenoid is a very important pigment in all business sector as it has many advantages to the human health. However, carotenoid available is being synthetically produced, which is bring negative side effect to the environment and human health. Thus, consumers seek for a natural carotenoid, which then lead to the utilizing of microorganism sources. Problem statement. Carotenoid available are being synthetically produced. Consumers are demand on natural carotenoids-based products for a healthy lifestyle. Therefore, the attention has been gathered toward the microbial production of carotenoid. There are three objectives to be achieved in this experiment. First, to optimize the pigment extracted using various solvents. Second, to determine the antimicrobial activity of pigment extracted. And third, to determine the hemolytic activity of pigment extracted from Rhodotorula mucilaginosa strains. Literature review. First, marine yeast. They can be divided into two obligate and facultative groups. Second, Rhodotorula mucilaginosa, also synonym with Rhodotorula rubra. The notable characteristic is the formation of natural carotenoid biosynthesis. Third is carotenoid. A pigment, mainly yellow, orange or red fat soluble pigment, can be divided into keratin and xanthophyll, being used as food colorants for decades and has many health benefits and also possess anti-inflammatory effect, antimicrobial and antioxidant properties. False. Antimicrobial properties of carotenoids extract from Rhodotorula genera. Major pigment produced are beta-carotene, toluene, and terulahodine. Studies have shown that toluene and terulahodine have strong antimicrobial properties against E. coli and S. aureus. Fifth, hemolytic activity. Hemolysis is a group of protein produced by certain microorganisms causing the lysis of the red blood cell membrane in the groove substrates. It has three types: alpha, beta, and gamma hemolysis. Areas and methods. Here are the flowchart of the experiment. For sample preparation, there will be two methods. The yeast will first be grown on PDA medium at 25 degrees Celsius for 2 days. For the first method, single colony will be transferred into YPD broth and incubate at 27 degrees Celsius for overnight. Then 3 ml of YPD broth will be pipetted for inoculation in semi-synthetic medium. Sample will be stored in shaker incubator at 150 rpm and 27 degrees Celsius for 72 hours. The second method will be inoculating yeast into 100 ml broth and incubate in shaking incubator with 200 rpm. The broth will be left at 25 degrees Celsius for 24 hours. Then the inoculum will be added into fresh 200 ml YM broth with 1% V/V ratio in 100 ml aluminium flask, and it will then be incubated in shaker incubator at 200 rpm for 144 hours. For pigment extraction, the fermented sample will be centrifuged at 8000 rpm and 4 degrees Celsius for 10 minutes. The biomass will then be dry at 60 degrees Celsius overnight. The dry mass will be mixed with DMSO and different organic solvent, which is ethanol, methanol, and acetone for pigment extraction. The mixture will then transfer into a fresh centrifuge tube and subjected to sonification at 50 kHz for 50 minutes. The mixture will be worked at maximum forces and left for 10 minutes before further centrifuge. Centrifugation process will be proceed at 5000 Sg and 4 degrees Celsius for 10 minutes. Next, the pigment supernatant will be collected to centrifuge again. Next, the supernatant will be collected and subject to evaporation at 60 degrees Celsius. And last, a solvent will be added to dilute the concentrated pigment before using in the antimicrobial assay. For antimicrobial activity assay, the microorganism will be grown in Crypticus soy broth at 35 degrees Celsius for overnight and will be adjusted to match a 0.5 McFarland standards before being used for antimicrobial activity assay. For pepper disc technique, 0.1 ml of inoculum will be sprayed on Mueller-Hilton agar, and then sterile antibiotic pepper disc will be sold into extracted pigment at different concentration. And after incubation of plate at 37 degrees Celsius for 24 hours, the halo of inhibition will be measured. Penicillin or gentamicin disc will be placed in the plate as a comparative standard. For determination of minimum inhibitory concentration, the pigment extracted at concentration of 2, 4, 8, 16, 32, 64, and 128 mg per ml will be mixed with sterile MHA agar followed by 0.1 ml of microbial suspension will be plated as pour plate. Incubation will be done at 37 degrees Celsius for 24 hours, and next the MIC of the pigments can be evaluated. To determine the minimum bactericidal concentration, the bacteria will be subcultured from the plate containing MIC to the nutrient broth. Nutrient broth will be prepared according to the protocol described by Ozoid. The mixture will be mixed and dissolved, and next will be sterilized by autoclaving at 121 degrees Celsius for 50 minutes. The MBC will be considered as concentration of the pigment that the bacteria did not grow. For determination of hemolytic activity, 1 drop of extracted pigment suspension, 5 times 10 to the power of 6 will be transferred to the sheep blood agar. The plate will be incubated at 37 degrees Celsius for 7 days and will be prepared in triplicate for each isolated. The presence of clear hemolysin in the medium will be considered as a positive result. For expected result, treatment with DMSO and ethanol will give highest yield of carotenoid compared to the other treatment. The carotenoid produced by the marine Rhodotorula mucilaginosa are expected to have some level of antimicrobial properties, and the presence or absence of hemolytic activity from extracted pigment will also be determined. The next slide will show the CAN chart and also the reference for this experiment.